Hello, everyone, and welcome to today's webinar, Candida auris, PCR Screening for Infection Prevention. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Diasorin Molecular. To learn more, visit molecular.diasorin.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. All questions will be answered following the broadcast via email. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Maura Pieretti, Scientific Director for BayCare Laboratories. Maura, you may now begin your presentation.
Thank you. Thank you very much to LabRoots and Diasorin for giving me the opportunity to share my lab's experience in dealing with Candida auris. I also want to point out that I'm not receiving any compensation for this seminar, and therefore I have no conflicts of interest to declare at all. So we will start by describing the impact of this relatively newly identified yeast and why this topic is so relevant and so serious. We will review identification challenges, review recommendations for screening algorithms. We will also go over how we implemented these algorithms. We will go over how we implemented our laboratory test implementation and validation, and review the data that we have accumulated so far. I'd like to start by reviewing a few facts about Candida auris. Let's start by saying that it first of all was identified in 2009, so it's not too long ago, and it was identified in Japan initially from a ear canal infection. The name auris actually derives from the Latin word auris, which means ear, and Candida auris quickly spread and was rapidly identified throughout the world. In 2011, in fact, the first bloodstream infection was identified in Korea. Candida auris tend to infect patients who are already ill or have other serious medical conditions, and such as people who have long stays in hospitals or in nursing homes. The mortality, unfortunately, that is associated with Candida auris infection is very high. In fact, about 1/3 of patients will end up dying, and that's very unfortunate. The other component of Candida auris, apart from infection, it tends to colonize patients. It has the ability to persist on the environment, colonize patients, and patients who have no symptoms are not affected by an infection. So these colonized patients can spread it to other patients, and therefore Candida auris is a true health-acquired infection, and it's becoming a really serious one. To add to the gravity of this pathogen, it is also very resistant to treatment, and some strains are resistant to all three classes of antifungal medications, azoles, echinocandins, and amphotericin B. And then the last leg of this threat is also the fact that this organism is not being very easy to identify in the laboratory unless very specialized testing and technologies were used. And of course, missed identification or wrong identification can equally lead to inappropriate therapy and also to further spread. You can see with all of these really grave matters why the CDC really classified this threat as urgent. And in fact, the CDC in 2006 sounded the alarm for this threat being classified as urgent. Only about 1 week later, the public health in England announced that it had identified Candida auris. in the WHO followed very soon after, warning about Candida auris, and then the European CDC Commission also sounded the alarm over this. Reports in the press, of course, add on to making this visible to the public eye, as it should, and cases began spreading in the U.S. Starting in 2015, more and more clinical cases of infections were identified, with a dramatic increase every year. This chart here, which is from the CDC, shows the over 300% increase that occurred between 2015 and 2018. Moving on to the spread, this global map here shows how the spread is really worrisome. This is, again, from the CDC website, and shows how many countries, over 30 countries in the world, including the U.S., have identified cases of infection and local spread. Other facts around this is that, of course, the cost to the healthcare system is particularly high. This is the cost that has been estimated for Candida auris infections in U.S. hospital, over $1 million. Bloodstream infections are very important and very grave, and they are those that tend to have the higher mortality associated with them. When I looked at actually the CDC website again just recently, this is showing again the map of the world where Candida auris has been identified, but specifically this sentence here made me really pause. This map is no longer being updated given how widespread Candida auris has become. And this was as of February 2021. So it gives you a little bit about how worrisome the situation is. What is also interesting is the fact that it appears that different Candida auris with different clonality have probably emerged in the world independently of each other. And this is at least according to data from whole genome sequencing. They're isolates from different parts of the world that actually belong to different clades. And as you can see here on this phylogenetic tree, you see, first of all, how closely related Candida auris is to other types of Candida, such as, for example, Candida humuloni. You can also see the 5 different clades that we now know as of today. And we know also that clade 1, 2, and 4 are responsible for invasive outbreaks by multidrug resistant strains. Clade 2 is the one that was first isolated in East Asia. Clade 5 is the most recent one with cases isolated in Iran, according to reports. So it is interesting that they have probably evolved simultaneously and independently of each other. The observation that different clonal lineages are distributed over the United States, for example, is really supporting the idea that these have been independent introduction with subsequent local spread inside the U.S. Moving on to other characteristics of Candida auris, it is a very hardy pathogen. And it is quite different from other Candidas. It can grow in the presence of high salt, over 10% sodium chloride, which is, again, different from other Candidas. It is also interesting that 2 studies have reported that in the presence of high salt, it actually can form some pseudo-hyphae like morphologies. And this suggests that this transition may be an adaptive modification that this organism is capable of doing when conditions change. Can live at high temperature, relatively high temperatures compared to other Candidas. In fact, it lives very well at human physiological temperatures between 36.5 and 37.5 degrees centigrade. And it can live up to 40 degrees and even 42 degrees, which would be really high fever. It is very resilient. It can survive on both biotic surfaces, but also abiotic surfaces. And in fact, viable Candida auris that can be grown in the lab has been recovered from bed rails, doorknobs, etc., where it actually creates a sort of a biofilm. And so this obviously, you can imagine how this can definitely contribute to the spread within the healthcare settings. So the transmission that has been demonstrated is definitely from human to human through contact, but also through environmental surfaces. And this occurs, as you can imagine, can occur in hospitals, especially when the there's a long-term care stay, or in nursing homes. So the associated risk for infections are the length of stay in specific units within the hospital, other diseases, an immunosuppressed state, and exposure to antibiotics, and age and devices, which we'll see a little bit more in detail. Let's now look at for a few minutes at the clinical presentation of the invasive infection. Again, I'm a laboratorian, I'm not a clinician, but I wanted to do a little bit of an overview on the actual clinical presentation. As mentioned, the infection very often occurs in people who are already ill, which can make recognizing the symptoms really, really difficult. It occurs in patients who have breathing tubes, feeding tubes, central venous catheters, patients who have previously received antibiotics or antifungal medications. The early symptoms can be fever, respiratory distress, and the lack of response to antibiotics. But it may vary very much with the primary site of infection. Candidemia, if it occurs, which is basically the infection in the blood, will lead to blood dissemination, and it can end up producing a generalized infection, septic shock, and multi-organ failure. It is therefore, CDC recommends preventing these type of infection, especially in people who have devices such as feeding tubes, breathing tubes, and catheters. Really, the adequate management of these devices is really important to try and limit the infection itself. So colonization, on the other hand, makes people mostly asymptomatic, but capable of transmissions to others. And importantly, they are also at risk of developing their own invasive infections, so especially if they're already ill. So while colonization clinically is less important, however, from a healthcare perspective, it's particularly important to identify colonized patients. So they need to be screened in order to be identified. More recent data, just to really point out clinical cases and colonization cases, and again, this is from the CDC, you can see how in the past year, this is 12 months worth of data from September 2020 to August 2021, there have been over 1,000 clinical cases reported and over 3,000 colonization cases reported. Also note the distribution of cases on the US map, which is not equal, so some states much more affected than other states. States in the Northeast, California, Texas, and Florida, as well as Illinois, appear to be the states with the most cases to date. To add to this really, really dire situation with Candida auris, of course, we also had the COVID-19 pandemic. And here, really, Candida auris became a lurking threat for a lot of COVID patients. COVID patients who tend to have long hospital stays and are already by themselves severely ill, where basically we had co-infections of Candida auris and COVID-19, SARS-CoV-2 reported, and a few outbreaks in several states in the U.S., including the one that I'm reporting right here in Florida that was published in MMWR in January 2021. So really a perfect combination of really high-risk situations for these patients who are staying in the hospital. I'd like to move on now to lab identification and all the challenges relating to lab identification. As I mentioned, the organism has not been easy to identify and has been often misidentified or missed altogether. There are 4 categories that I'm going to be going over regarding lab identification: phenotypic, biochemical, and MALDI-TOF, and molecular identification. The challenges have been that because Candida is so closely related to other types of Candidas, it has been misidentified as those. And also the other challenge is that it is not common practice in the laboratory to speciate all Candidas unless they are identified in infections from sterile sites. So there are about 40% of clinical cases are not from the blood, are, for example, urine and bile. And in general, laboratories have not been in the practice of speciating Candidas to the species level when it comes from non-sterile isolates. So they would basically just be reported as Candida at the genus level only. Obviously, in a healthcare setting that has Candida auris spread, this will need to change, and it's recommended by the CDC. Phenotypic identification, what we mean by this is basically looking at colonies on a plate, on growth plates, and there have been many challenges because it's very hard to identify and very easy to misidentify. What I'm showing here are actually some specialized agar plates, the chrome agar plates, which are not the plates that are commonly used in most laboratories. They can help somehow in the identification, but even with these plates, the colonies are really hard to tell apart from other types of Candida. As you can see here in the left picture, Candida glabrata is purple, tropicalis is blue, and auris is white, however, not very easy to distinguish. And then also because in some cases, as you can see, a pure colonies here of Candida auris on the middle plate, and even in those cases, the color morphology can vary from white to purple. So bottom line is that phenotypic identification can absolutely not be the sole method of identification for this organism. The biochemical methods are also a little bit fraught with errors. I'm listing here in this table various types of commercial systems that are used in a lot of clinical laboratories, at least in the US. And all of them pretty much have had trouble identifying Candida auris. As you can see, Candida hemuloni could be ID'd, and in fact, when Candida auris was actually present, and other types of Candidas. This is changing, fortunately, recently. However, this has probably led to a lot of misidentification just in the past, and probably still is if these systems have not been updated. Fortunately, matrix-assisted laser desorption ionization time of flight, more commonly referred to as MALDI-TOF, mass spectrometer, has really come to help. These systems, and I'm showing here the 2 major systems that are available, at least in the US clinical lab, the Bruker and the bioMérieux, are now both capable of correctly identifying C. auris. However, it all depends on the database and the library that is loaded on these instruments. If the software has to be the IVD Claim 4 and more recent for the Bruker, and it has to be the IVD library version 3.2 or more recent for the bioMérieux. Prior versions of the software was actually not identifying Candida auris. So good news for those who have mass spec in their clinical microlabs. But again, the right software and the right library need to be added here. And then it comes to molecular methods. And of course, molecular methods identify, of course, the genetic material. In the case of Candida auris, ribosomal DNA seems to be the preferred target for both sequencing and amplification-based methods. And this is reported in a variety of publications. For sequencing the D1 and D2 region of the 28S ribosomal DNA, or the internal transcribed region, ITS, so also the ribosomal DNA, can accurately identify Candida auris. And the reason are a few. It is a very good region to target here because it is universally represented in fungi. It has multi-copy nature, so it's easier to pick up. So there are these really well-conserved regions, but they're also interspaced with highly variable regions that allow for differentiation of closely related species, as you can see in the graph here at the bottom of the slide. So obviously, this is a very good way to conclusively identify Candida auris. Of course, our DNA sequencing is not available in all labs. Amplification methods, on the other hand, are a little bit more available. They target the same regions, and also they can bring to the same precise identification of Candida auris. I also want to mention here the GenMark ePlex blood culture ID fungal panel, which to my knowledge is the only one test that is actually an FDA-approved molecular test in the US for the identification of Candida auris. However, obviously, this is just for positive blood cultures. That's how the panel is meant to be used. So I wanted to mention that as well as an extra tool. When it comes to molecular methods in general, they all rely on amplification, as I said, of the genetic material. They come in many flavors. Of course, there are many different types of molecular amplification methods, different types of PCR or other type of isothermal amplification. And of course, the big winner seems always to be real-time PCR in terms of how frequently this is being used in the literature and how frequently this is implemented in laboratories. A few considerations as my lab was thinking of adopting an amplification method for the identification of C. auris is, of course, what are the important characteristics that we're choosing, that we are wanting in this method. So obviously, we want the method to be sensitive, specific. Of course, that will depend on the primer sequences and the probe location. What other equipment is already available in the laboratory, so that we don't, obviously need to purchase something specific just for one new test. What type of clinical samples do we need this a molecular method for? Is it for the identification of positive colonies or for the identification of colonization? So very different clinical scenarios. Of course, also cost is always a big issue in laboratories these days. And how much cost is lab really willing to absorb? As you know, if you added a new identification method, not necessarily you get more reimbursement all the time. So what can lab absorb in terms of cost of setting up new things that help in the diagnosis of disease or the on screening methodology, but what can really be done? And also, of course, it's the turnaround time that is needed is very important, choosing the methodology. And also, as you can all imagine, what else is going on in the laboratory. As my specific experience, we were in the process of considering implementation of C. auris molecular methodology, as we will see later. And we started thinking about this at the end of 2019, actually probably earlier than that, but started making the final plans for actually going live at the beginning of 2020 with this new test, until, of course, COVID happened, and we had to put everything on hold and deal with the more urgent threat at the time. As a conclusion to the review of the laboratory methods here, before I move on to my own experience with implementing this, I've mentioned the fact a couple of times that Candida auris seems to be very resistant. So I wanted to take a minute to look at susceptibility. And first of all, remind that the CLSI guidelines recommend to do complete susceptibility studies for Candida auris that are isolated from an infection. I, however, also want to remind you that there are no established breakpoints to date, and there's no correlation, final correlation between the breakpoints and the clinical outcomes. So this is obviously data that will be changing. I'm hoping that CLSI will update very soon, but this is the situation at the moment. And these are the three classes here of antifungal medication that are commonly used, and you can see that unfortunately the main drug, fluconazole, and this is the azole category, Candida auris is over 90% of Candida auris is resistant to fluconazole. So obviously this is not a drug of choice. 30% of the isolates are also going to be resistant to amphotericin. And so with the last class of antifungal medications still showing the least amount of resistance, so probably being the drug of choice in the majority of cases here. Okay, so now I want to move on to my specific experience, my lab's experience with the Candida auris. And Candida auris was spreading in Florida as early as 2017. It was first isolated in the southern part of the state, with the first few cases in 2017 in the Miami-Dade counties, mostly with local transmission within and between healthcare systems. More recently, however, we've had cases also in other areas of the northern and western parts of Florida. And as you can see, the symbol here, BayCare, my healthcare system, is located on the west coast of Florida. So a couple more words specifically regarding the BayCare healthcare system for which I work. And I'm showing here sort of a blow-up of the Tampa Bay region. You can see that the Tampa Bay region is located on the west coast of Florida. Our healthcare system covers a pretty large area. Tampa Bay region is home to almost 4 million people, and BayCare healthcare represents almost 40% of the healthcare for this population here. What I'm showing here is all of our different hospital labs, so that each microscope here represents a lab in the area. And our hospital labs are pretty sophisticated. We have a lot of decentralized testing when it comes to molecular testing, a lot of PCR testing, a lot of COVID testing is actually done at the hospital labs close to the patients. We also have, however, a central core lab, which is indicated by the star here, the red star in the map. And the central lab has a core microbiology laboratory, a core molecular laboratory, and other areas of specialized testing such as cytogenetics, flow cytometry, and so forth. So we came into, I also want to mention here, before I forget, that the core microbiology laboratory is fortunate enough to actually have all three systems that can now accurately identify Candida auris from positive cultures from infections. We have both mass spec instruments as well as the VITEC 2 software version 8.01, which allows for the specific identification of C. auris. It is true that with this system, just as a word of caution, if other Candida, such as Hemiloni, for example, are identified, it is still possible that Candida auris may be present, so more studies or more identification with the mass spec may be needed, but it can identify C. auris at this point. So as I mentioned, we started seeing Candida auris cases in Florida, and we actually reported and recorded the first case of infection within our healthcare system exactly in March 2020, which of course all remember as the beginning of testing for a lot of our laboratories for SARS-CoV-2. This patient had been transferred from a long-term acute care facility, and we identified it and immediately started a screening protocol for identifying colonization. So we recognize the threat. Unfortunately, the lab at that point was not able to do any testing in-house. However, screening for colonization and identifying colonization of patients who have been exposed to infected is particularly important. So I want to go back for a second to talk about colonization. So colonization occurs in those people who have C. auris but are not infected, are asymptomatic. Unfortunately, these patients can remain colonized for months. And these patients can spread it to others, but are also a risk to themselves. Also, importantly, my infection prevention colleagues tell me that there is really no good way at the moment to decolonize patients. And so there's really, despite trying with baths, with chlorhexidine and other agents, it's really almost impossible to decolonize them completely. So where do people get colonized? They can get colonized in the mouth, the rectum, the urine, the nose, the ear, the external ear canal. However, it seems from consensus in the literature that the best spots in the body where people are always colonized and seem to be the best spot for collections are the axilla and the groin. And so these are the sites that are actually being collected when somebody is tested for colonization. And whom to screen, there are a lot of publications around this and a lot of recommendations. And of course, whoever comes in contact with an infected patient. Also, the recommendations are to screen patients who are admitted to the hospitals who have a history of healthcare stays abroad, patients who are colonized or infected for carbapenem-producing organisms, and patients who have a suspected exposure. So based on these recommendations, we started our own screening algorithm, and infection prevention here at BayCare was kind enough to share their algorithm with us, as we've been very close collaborators in this, as many as other projects. So Candida auris, of course, is subject to contact bleach precaution if somebody is infected. And so there are many signs and many things that need to be taken into consideration when treating these patients. We put in charge the infection prevention team to identify patients and gave them some tools, such as what we call them a standing order for patients with a history, previous histories of C. auris. As I said before, patients with a history of carbapenem-resistant organisms, roommates, and so forth. And so infection prevention has been in charge of screening patients who are at risk of being colonized, and we have developed very precise guidelines on what to use. We're using nylon flux swabs. Those that come in a white cap with liquid amine seems to be what is also being used in the literature from what I've seen, and we're instructing everybody to collect both the right and left axilla, and then with the same swab, collect the right and left groin, and then bring these to the lab. As I mentioned previously, we were not able to do these in the lab immediately, and so we started by actually sending our screening tests based on this algorithm to the closest lab, the part of the network, and the closest for us was the Tennessee State Lab, which was great in that they were setting up cultures and PCRs for us. And however, due to the distance in transportation, the paperwork involved, obviously the results were not back in a really, really timely fashion to really quickly identify colonized patients. We decided that we needed to move on and start looking at molecular methods inside our own central core laboratory. We ended up developing a laboratory-developed test, and we had already quite a bit of experience with ASRS, specifically ASRS from Desorption, as well as other vendors. And we looked at our options, and we decided that this was a good combination of reagents available. The primer pair in the ASR fundiasorin targets the ITS2 region, is labeled with FAM. We also knew that we needed other components, which we were already very familiar with, such as extraction and amplification reagents containing internal control, DNA, and primers to control for PCR inhibition. We also knew that, and we had experience with temperature-activated master mix, which allows for direct assay that do not require a separate extraction. Eventually, and I'll talk about this in a minute, we ended up also investigating this fungal lysis solution from diazorin, which ended up being very helpful. And we knew that there were several control materials available from a variety of vendors as well as CDC. So we set up to set up our own assay, and actually we tried quite a few things because we really wanted this assay to be relatively quick, fast, relatively simple to set up, especially since we were really busy with doing thousands and thousands of COVID tests. So we tried first the direct sample approach with no extraction at all and putting the sample directly into the PCR master mix. And this obviously was very fast, but it was really not quite as sensitive as we wanted it to be. We tried a couple of off-board extractions, and of course, this is not a very extensive list. We decided that this was also not really adding a whole lot to the sensitivity, and the sample just didn't seem to lyse sufficiently with the methods we tried. We could have tried a few more things, of course, but then the fungal lysis buffer came available from diazorin. So we tried this, and it actually proved to be the winning ticket. The fungal lysis buffer allows us to basically lyse the sample ahead of time by adding some volume of the sample with about 1/10 of the volume of lysis buffer. The sample pretty much cooks for 30 minutes at 60 degrees centigrade, where it lyses quite efficiently. It's then deactivated at 95 degrees centigrade for 10 minutes. And then it's basically ready to be set up in amplification, where we're using for many different real-time PCR instruments could be used here. We're using the diazorin liaison MDX and the 96-well direct sample plate, using 2 microliters of samples and 8 microliters of master mix. So as I said, we tried a few things, but this seemed to be working pretty well, and I'm showing you here some examples of results where you can see the amplification curves. I have 4 samples here, to our patients and to our controls, and you can see that the amplification curves of the first patient is just purple. That's just the internal control. The amplification curve of the second patient is actually both the green and the purple curve indicating amplification of the target for C. auris as well, and these are the negative and the positive control. So after we finished with sort of these finding out what the best conditions were, we could do our validation studies to be able to bring this test live in-house. As I'm sure a lot of the laboratorians here in the audience are familiar, validation studies, of course, include accuracy, precision, sensitivity, and specificity. For accuracy, I must say it was relatively easy for us because we were already sending the test out. We were already swabbing a lot of patients and sending them out to the Tennessee lab. So we basically compared our PCR to the Tennessee lab PCR, and also we had some reference material and known concentration that we could also use. For precision, we wanted to see how reproducible the study was over different days and different technologies, and we looked at specifically at the coefficient values for reproducibility, for looking at the CT values of the positive control. For sensitivity, we were able to get tighter positive cultures at tighter concentration and dilute those down and do our limited detection studies. And for specificity, we looked at the performance of the assay against different types of candidas. and other types of organisms as well. And the results were really good, and with 100% accuracy, really good reproducibility, and a limit of detection that we could prove to be 2 colony-forming units per reaction. So we were able to go live with this test and introduce it in-house at the beginning of this year. That was a major win because it allowed us for much more rapid results, of course. And we were able to now introduce the test in the EMR, allow the infection prevention team to, with just a few clicks, identify the patients, put them in isolation, and order both the culture and the PCR. We decided to go live with both tests being done on the same swab, again, collecting the axilla, groin, one swab, and setting up with the same swab both tests. This allowed for much faster results, and everybody seemed to be very happy that we were able to do this, including the lab, who had to delay the project for a while. So the volume of testing has increased over time, and this is showing real data from when we went live to pretty much last month. At one point, we decided we wanted to really look at the concordance between the PCR and the culture, because we really were doing a lot of work and a lot of screening, and we wanted to see how much value the culture was really adding to this. So we compared it and saw that for the culture, we actually had 4 patients who were positive by PCR but negative by culture. And in a way, this was sort of expected. And we were able to confirm that these cases were actually proven to be true positive based on their history or subsequent cultures that were then positive. On the other hand, though, the culture was not really identifying any additional positives. There were 0 that were PCR negative and culture positive. So we decided to bring this up to our collaborators. We tend to make decisions here when it comes to screening, especially in other testing in the lab, really in a collaborative fashion. So we discussed this at our infectious disease collaborative with the physicians and as well as infection prevention. And we decided that it was really time to stop doing both cultures and PCR. And so we discontinued that in August and are proceeding just with a PCR test as our screening test. To date, we have screened over 1,300 patients, and I'm sure we will continue to do so and probably increase the screening even further. It is a daunting task to have one more test with such high volume. However, we feel this is really important for the system. We also really keep a very close eye on the turnaround time of the results because obviously these patients are in isolation until proven not guilty, so to speak. And so we really keep a really close eye on the data. And here I'm showing the volume of testing in each month, and you can see that it's going up and down, but mostly up. In some cases, there are some events that prompt more screening. And I'm showing you the turnaround time from the in-lab stamp to the final result, the verified result, which is important, but also as important and more important is the collect to verify turnaround time, which hovers around 30 hours. At the moment, we're batching once a day, and we're not doing much testing over the weekend, but we're already thinking that we want to do a little bit better than that, and probably we start testing heavily on the weekend as well. And keep in mind that we're doing this in a central lab location, so there's some transportation for some of our samples coming from our hospitals. We are the radius of the BayCare health system is pretty wide in terms of mile and transportation. So at one point, we may need to decentralize this test and put it closer to the patients in the hospital labs. We're not quite ready to do that now, but something definitely that we need to keep in mind. Another important thing, to consider is the fact that we need to give better tools to our clinical teams to identify patients. So we cannot just rely on infection prevention identifying these patients, but we need to make the emergency department, the ER team, in charge of identifying these patients when they first come into the hospital. So now we've introduced other tools called the Power Plan, where questions are being asked at the triage, whether the patients have had a stay in a long-term acute care hospital, or they're coming from a facility that has known cases of C. auris, and there's active DOH, Department of Health communication, on a list of facilities that have those. So if the questions are answered yes, then the PCR test is actually ordered automatically, so that we don't miss any potential colonized patients. So having said all of this, I want to conclude with a few thoughts. I think I've imparted on all of you that Candida auris is a fungus that has very, very serious consequences and is spreading in the healthcare facilities and in the U.S. as well as the rest of the world. Accurate identification of this organism was not always possible in the past. However, new methodologies are available, new software, so accurate identification of both infections in the positive culture as well as colonized patients is now possible. Specifically, rapid identification of colonized patients is really necessary and needed to prevent hospital-based transmissions. Molecular-based methods are really ideal for screening because they're fast and they're very convenient, and of course, it's even better when they're available in-house. And the last thought I have is that really for all these projects, as we've learned through COVID, but also through with this specific project on Candida auris, that interdepartmental collaboration is really vital to make things successful. Really, lab cannot work in a silos, and so we work very, very closely with infection prevention, infectious disease specialists, and the hospital staff. And on that note, I want to thank my collaborators at BayCare on this project, the lab supervisor who did really spearheaded all the validation studies, the technical director, Dr. Seferi, the technical director of microbiology, the director of infection prevention, Suzanne Chandler, Dr. Gupta, who's the quality and project improvement director. And of course, thank you also to the DiaSorin molecular team.
Thank you, Maura, for your informative presentation. We would also like to thank LabRoots and our sponsor, DiaSorin Molecular, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.